Good morning, everyone, in Woods Hole and out in the world. We are very glad that you're still here on day 3 of this workshop. We first would like to thank you for the very hard work you did over the past 2 days, thinking about science objectives and putting them together in the synthesis slides. For those who have not finished the slides, you have until lunch today. Right? Heather, these are the instructions that I'm getting. So today we are switching gears. We all know now the science objectives, right? So we can go to the implementation. And to start off the implementation, we have a few presentations, and we will start with Ben Twining from Bigelow.
Okay, great. Good morning, everyone. There's Advance. Awesome. Cool. So yeah, it is great to be here. It's been wonderful to be in a community where I feel like, or in a meeting where every single person I have an interesting conversation with, which has been a treat. And as Ingrid said, now our goal is to try to start thinking about how do we get to how? How might we sort of implement these and test these ideas through a BioGScapes program? And so my charge to start this morning is to channel my inner Philip Boyd. He might actually sit inside me, so he's not out there hearing my jokes, unfortunately. But yeah, Philip wanted to sort of look back at what he's been calling sort of proto-BioGeoscapes field and lab studies to look at some of the shortcomings and how we might try to tie them together using some approaches. So yeah, any ideas here that are good are credit Philip, and the shortcomings of communicating them credit or blame me. And I want to also recognize a whole host of collaborators and colleagues who have provided slides without studies that I'll show, and daunting to give a talk with other people's slides in a room where they mostly are. But we'll see how this goes. Okay. So here's sort of what I'm going to break the talk up into 4 chunks. First, starting with sort of a brief survey of some recent, I got survey in there twice, a brief survey of recent field programs, both biogeochemical and sort of omics oriented. Then step it back to think about how we might fill some of the gaps in those survey programs through maybe repeat occupations or lab studies. And then move back into looking at some of the previous studies about sort of how some of those have done that, in particular using manipulations at sea and trying to combine omics and biogeochemistry, and then try to summarize some challenges and potential solutions. Okay, so one way that Philip and I have been thinking about this is sort of plotting programs on a sort of a 2-dimensional matrix of discipline and approach. So here on the x-axis, you can think of moving sort of from biogeochemical programs to maybe omics programs and having space in between. And on the y, moving from sort of a survey mode to a process mode. And again, lots of space in between. And so I'm going to go through some of these previous studies and talk a bit about, sort of summarize what they did to sort of give you a flavor of what has been tried. And I will hopefully use a little box. We can't see that. Can we move our little Zoom window up there? Can I make that? Will it go away if I hit a magic button?
Not yet.
Not yet.
Technology is not there.
Yes, I'm not moving from this spot. Oh, yeah. So GeoTrace is, as a sort of example of a biogeochemical survey program, I think everyone's familiar with this. This is an example of data from the North Atlantic transect, the U.S. transect GA03. Got a little bit, there we go, is our pointer. Moving from North America to Western Africa. Many trace elements and isotopes measured. Here, just showing examples of dissolved iron and particulate iron. This is sort of a survey approach for biogeochemistry. And GeoTrace certainly has had, and I'm going to show a number of the process studies, but the bulk of, I would say, the GeoTrace's program and what's really been pulled together in an international way has been the surveys. And these have included measurements of nutrients, very high quality measurements of nutrients and pigments, carbonate system, oxygen, so some of the other biogeochemical sort of parameters that have been discussed here. But it has very much been a snapshot, right? The measurements we're getting are season specific, and that limits our ability to sort of draw biogeochemical or really biological inferences from them. I want to mention that there have been some biological samples taken on them. On this particular cruise, my group took single-cell samples for quotas. So here we've got iron to carbon ratios and cells moving from west to east. And as part of what was then called the sort of biogeotraces sort of effort, there have been a number of molecular biological samples, for example, single-cell genomes, collected across this transect as well. So there is some biology sort of, we're scratching over on that side, but primarily this has been a geochemical survey cruise or approach. So here, if we think about going to the right side of our x-axis over to the omics side, Terra is a great example of this. Everyone's very familiar with Terra and all that has come from it from all this omics data. I'm not going to read all of these, but we know millions, tens of millions of genes, organisms, protists, interactions, a massive amount of biological data that has come from Terra. And if you think about the sort of questions that can be addressed as you sort of move up to the right on this figure, is moving from the genes to the organisms and trying to take it all the way to that ocean biome and the biogeochemical and climate questions. That's required modeling for sure, and so we need to be able to get this information into models. But it really requires then connecting the omics to biogeochemical rates and biogeochemical mass balances. And in an effort to do that, or sort of to make this connection, here's an example of that. This is a paper by Caputi et al. Looking at sort of the interactions of iron and the metagenomic and metatranscriptomic responses of communities. And it became apparent in this effort that there was not concurrently measured dissolved iron on the cruise. And so iron measurements or iron predictions were taken from a global model. You can see here, these are sort of a map showing the metagenomic and metatranscriptomic data sets with the color coding showing iron. So, very strong predictions of iron gradients. But without those concurrently measured iron concentrations, the authors really chose to focus on an area near the Marquesas where you have a downstream stations near an island. So you get a presumed island effect. And these showed, strong shifts in the metatranscriptomic and metagenomic communities, and the ICIP and other proteins responding to those. But again, it suggested this sort of natural iron fertilization effect, but it was difficult to really tie it up in a nice bow without that measurement. So we think on our x-axis of biogeochemistry and omics, the BioGoship project probably sits sort of in the middle between these flavors. This is a relatively new effort to add biological and omics sampling to the GoShip repeat hydrography program in order to link marine biodiversity and biogeochemistry. And this figure shows the sort of biological measurements that have been added to this program: flow cytometry, imaging, optics, omics, and particulate measurements in order to try to answer some of these questions. And a nice example of the kind of information you can get from that global scale approach of connecting these is this paper by Ustic et al. In 2021, where they look at nutrient stress gene transcription in Prochlorococcus across these global or these basin scale surveys. And you can see genes that are associated with iron or phosphorus, nitrogen, or mixtures of those limiting nutrients, and how those genes are expressed across these environmental gradients. So showing really the power that you can get by combining the omics and the biogeochemistry. Okay, there have also been process studies that have been done both in the biogeochemical and omics space. And so here I'm moving to the upper left corner of our space and give an example of Iron Cycle 1, which was a process study in unperturbed low iron waters. So the team went out and marked a patch of water with sulfur hexafluoride, just like the mesoscale ocean fertilization experiments had done, and then just watched and measured what happened with iron cycling. And by focusing on a constrained volume of water, they were able to look at, for example, changes in dissolved iron or dissolved iron ligands over time. And it's interesting to see, keep losing my pointer, interesting to see, for example, that dissolved iron is these profiles are relatively constant over this week-long period. But at the same time, the team was measuring iron uptake and iron recycling rates. And that allowed them to put together this budget that shows that these recycling fluxes are basically an order of magnitude greater than the fluxes in and out of that body of water, and showing the importance of these biologically mediated processes within this system. And that was really a new view into the fluxes of iron that was enabled by this process study. This was followed subsequently by the creatively named Iron Cycle 2, which also created an iron budget, this time during a spring bloom, again in low iron waters in the subantarctic. And so here you can see chlorophyll going up. You see this diatom bloom occurring in austral spring. That led to a rapid uptake of dissolved iron from the water column. And again, because we're looking at a controlled volume of water, we're able to not only look at uptake, but also look then at the type, basically the source of that iron. So here, the group or the authors have delineated water column integrated iron uptake to sort of uptake of new iron coming from outside of the system versus recycled iron, and have also been able to look at iron uptake by just a component of the community, in this case the diatoms that were blooming, and it's interesting to see it's a relatively small part of that uptake. And the team went further and looked at sources of regenerated iron. So here they're looking at herbivory or bactivory, fecal iron, viral lysis as processes that release that iron. So, this is a big step forward again in sort of getting to a mechanism of this micronutrient cycling. But without omics, there are very few details on microbial strategies or microbial community structure that might really help nail down those mechanisms. There have also been surveys deeper in the water column. I want to emphasize that some of the work's been done in the mesopelagic and below. This is some data collected as part of the proteomes project led by Max Sato's group, and here in a paper by Jackie Saunders. Here they're measuring proteomics across this area where they found about 80,000 proteins and looked at their relative abundance across these biogeochemical gradients. I think that'll go away, right? And this allows us to look at the types of proteins there and give an insight into how communities are responding to these gradients. On the biogeochemical side, also thinking about mesopelagic processes, the Peacetime project, also led, I think, by Phil Boyd and a paper by Bresak et al. Here, looked at subsurface iron remineralization, regeneration, and recycling from particle assemblages in areas that are dominated by biogenic particles compared to areas dominated by lithogenic, so very different particle types. And this had a big impact on the amount of iron released and the state of that iron, as well as the production of, for example, ligands by biology. And you see very different processes going on in these areas that tell us something about the regeneration of this nutrient. But again, without the omics, it's just sort of a black box of what's driving these biologically mediated processes. Okay, that's my little sprint through some of the sort of early survey field programs. Now I want to talk about sort of trying to fill some of those gaps that the snapshots provide by using repeat occupations or lab studies. So a biogeochemical example of this is the Bermuda Atlantic Iron Time Series study, or BATE. Very good at acronyms. And this was an attempt to measure iron cycling over a single year across 4 seasons at the BAT station. And by going multiple times and sampling the same system, we're able to constrain, for example, inventories of, on the left, dissolved iron or iron-binding ligands. And by combining those changes over time with measurements of particles and other forms of iron, and for example, colloidal iron, which is also plotted here in the center, we were able to test the sort of longstanding mechanism of ligand equilibration as being a control of dissolved iron, and understand that actually it couldn't explain the behavior of iron. And it required a new mechanism, in this case, sort of colloidal precipitation in interactions with carbon to explain the distributions of iron. And this was referenced yesterday as well. And so here, by not having just a snapshot and assuming what happened before and after, but by going back repeatedly, we were really able to pull out a mechanism of iron cycling. Sort of thinking about it from the biology side and the value of repeat occupations, here's some data from the SCOPE project, where they looked at microbial dynamics through repeat occupations. In this case, they're going across a gradient in the North Pacific of productivity from oligotrophic waters up into the more productive waters to the north. And on the right, sort of hard to see those labels, but this is Prochlorococcus abundance and Synechococcus abundance over temperature, sort of a proxy for latitude here. And you can see there's this gray shaded area, this hot spot where you see dramatic changes in the abundance of these important cyanobacteria. And because they came in multiple years, when they went in 2017, you can see, where's my cursor, this dramatic difference in Prochlorococcus abundance in that year. And they tried to sort of explain that through a number of environmental parameters, could not do that, and in the end came to the conclusion that it was actually the cyanophages and viral lysis that was driving this change in the community. And that was, here on the bottom plot, we've got cyanophage abundance versus total picocyanobacteria. And so showing the value of repeat occupations from a biological perspective, because by knowing what happened before, it allows you to make a new interpretation of what's happening when you're there. So a lot of value there from repeat occupations. Of course, there's also great value in laboratory culture studies. And these allow you to manipulate the conditions, allow to look for the response of microbial organisms to these forcings, which is hard to do at sea again. And so an example of this is from Boyd et al. 2015, where they took a Southern Ocean diatom, this is a Pseudonychia, and they exposed it to a range of climate change variables, temperature, CO2, nutrients, iron, light, and looked at the responses of first a number of sort of geochemical mass-based parameters such as chlorophyll, carbon, nitrogen, phosphorus, silicon, but then also connected that with proteomic measurements. And on the right, this is showing sort of a multidimensional view of the changes in the relative abundance of over 1,500 proteins and looking at how those responses group together. And you can see that the conditions, map together onto each other. So there's common physiological responses to these. And by having not just the sort of the elements on the left, these currency converters, but also the proteins that tell us about the mechanism, this is the way to connect and these trait space differences back to field studies, I would think. Okay, another example of sort of culture insights from culture. This is work on Synechococcus isolates responding to temperature and iron, work done in Dave Hutchins' lab. And here there's two different strains of Synechococcus, an oceanic and a coastal strain, looking at the response of both growth rate and iron and phosphorus content to temperature and iron availability. We see taxon-specific responses and taxon-specific responses to these multiple stressors. But again, by not just stopping there with sort of the sort of elemental response, but looking at the proteomic response on the right, these are relative or changes in abundances of oxidative stress proteins and metal chaperone proteins, also showing some strong differences between the taxa or the strains and the conditions. So again, connecting mechanistic information, physiological information with sort of our currency converter of the elements. Okay, so those are ways that we might think about, how could we then think about combining the value of those lab incubations with the value of working with natural communities. So sort of the plug here is for manipulations at sea. An approach to link the lab and the field is to try manipulation experiments. And so I'm going to finish with a couple of examples of that. This is one work done by Gilbert et al. From Steve Wilhelm's lab, working at the SOT station, again in the Southern Ocean. And in this project, water was collected from the ocean and then basically either titrated towards iron limitation with the iron ligand desferrioxamine, or titrated towards iron abundance by adding iron. So we have all these conditions shown in the sort of the color-coded gradient of iron availability. And what that allows you to do is sort of, prod the population in both directions. And you can see here on the right, here we've got sort of the control in the middle, added iron on the right, added DFB on the left. And you can see that growth rate responds to this, not just in a sort of a binary response, but in a gradient response, as does photosynthetic health of the organisms, allowing us to interpret sort of where the natural community was in that trait space. But again, by not just stopping with that sort of bulk analysis, but then adding, in this case, transcriptomics or metatranscriptomics on top of it, it allowed us to look at the responses of, in this case, individual ecotypes of Synechococcus. So here's, a taxonomic tree based on the RPOB gene. And there are Synechococcus ecotypes that classify or group together based on their response to iron. So this heat map is showing ecotypes that became more abundant with iron added versus those that actually became more abundant with iron removed, showing their different responses. So really, again, mechanistic, detailed ecological information coming from these manipulations that I think really helps us look at this trait space in a natural community. Another example, this is from Scope Gradients. I don't have data to show here, but this is a big project with many folks that is not only going across gradients, but then doing manipulations at sea where they are doing incubations for both top-down and bottom-up controls, measuring elements, but also omics and rates, and then incorporating it into models. So a nice example of that. And the last one I want to focus on is the ZipLock program, led by a number of British colleagues that, again, combined a survey, in this case across the North Atlantic Ocean, around 20 degrees. And along this section, you see significant changes in phosphorus abundance. So this is phosphate going down from west to east, whereas dissolved organic phosphorus increases. You also see a gradient in dissolved iron changing over there. And so the question was, how does phosphorus acquisition and phosphorus acquisition strategies change over this gradient? And so one way is to use that survey approach, in this case looking at proteins abundances for phosphorus, nitrogen, or iron or zinc uptake. And they saw differences, for example, in Synechococcus FOA, so one of the alkaline phosphatase proteins, versus Prochlorococcus FOA. Both of them show a gradient along that DOP gradient, which was really intriguing. And but interesting, we don't see a gradient, for example, in the iron biomarkers, despite that threefold gradient in iron. But I think the real power here is not to just stop again at this survey view, but to then do also incubation. So they did a whole complicated suite of matrix of incubations involving additions of zinc and iron, both cofactors for alkaline phosphatase proteins, as well as nitrogen and DOP, and then looked again at the response both in an elemental and in a proteomic sort of way. And interestingly, while there was no significant increase in growth rate or alkaline phosphatate rates, there were significant changes in protein concentrations showing interactions with iron and zinc as sort of was hypothesized. So showing these complex responses that are specific to individual groups, which will really, again, provide mechanistic understanding. Okay, how are we doing? Oh, we got a couple more minutes. So to try to summarize this, some of the challenges that I think these survey cruises show us are that voyages have limitations. There's only so much ship space on the ship, there's only much wire time, and so choices have to be made, and this is what we need to grapple with as we start thinking about the how today. And I would argue that intercalibration, broadly implemented and defined, really has benefit because it enables separate studies to be combined together, right? I don't think that our answer here is to have a single model of experiment or cruise, but by intercalibrating amongst the measurements, we can combine differently designed cruises together to come up with a global understanding. There's another challenge of linking surveys to time series and lab studies. And our sort of suggestion here is to consider common metrics or manipulations that might be done across multiple lab or field studies. For example, something like a biotitration or addition experiments that might allow for similar tests of trait space across studies. Towards the challenge of linking omics to biogeochemistry and metabolic models, as we've talked about, we need these currency converters. I think the challenge here is to agree on maybe not just a common set of converters, but even just an approach to how we develop those, because they're likely to be different by system. And finally, and this is I think something Phil is really excited about, is thinking about rates are one of those converters, but the tools we have to measure rates, as has been discussed here already, are pretty limited, low throughput, and prone to different results. I think we've also discussed. And so how can we develop newer, higher throughput approaches to do that? So thinking about next steps, I think thinking about a common set of manipulations, again, that we might consider. And I think we haven't talked much about what I like to call the franchise model. I don't know if anyone else is going to talk about that. But the idea that we can have common measurements or common manipulations that will allow us to pull together a bigger picture. Thinking about how we adapt the learning of these earlier studies to the themes of this meeting. And here I've sort of plotted the three themes, shortened a little bit, across, sort of onto this trait space. And I'm not sort of trying to say that this is where they fit in that trait space, but I think there are overlaps there that we should think about where they fit. And in particular, try to think about where those sweet spots are that link the themes of the workshop with the approaches that we can do. And I don't think there's just one spot where that sweet spot exists. I think for different questions, there will be different approaches that will give us the answers we need. And the last slide is just to encourage us to recognize that I think biogeoscapes can and should exist as an ecosystem within other ecosystems of programs that are going on. You know, certainly I talked about BioGoShip. There's other international large programs such as the NASA PACE project, a SCORE working group on changing ocean biological systems. There's the Biogeochemical Argo program. GeoTraces continues. There's a number of other large programs that are going on. And so I think our challenge is not to replicate necessarily what's in those, but to serve as sort of an ecosystem that connects them. Thank you. That's it. Okay.
Thank you very much.
You're welcome. Yeah. Oh, I don't get to talk. It is.
Hi, Ben. Thanks a lot for that presentation. I wanted to make a comment and then ask a question, I think. One of the things that is missing from this presentation, or what I should say is your presentation is focused very much on metal-oriented field studies. And there are at least 2 very organic geochemistry-focused field studies that were missing from your presentation. So Deep DOM, which was a cruise that built on the lessons we learned in GeoMix in 2013, and Bioscope, which is led by Craig Carlson out of Bermuda, and it's been going on since 2016. And so my question, I think it's a question, I want to just first of all put that out there, that the organics are missing from this presentation. But I'm concerned about a more general absence of organic geochemistry from some of these discussions. And I wonder if it has to do with the methods not being as advanced because we didn't have GeoTraces as a foundation, and so on. But I'd like to sort of have you comment on where there could be disciplinary connections that a group like BioGeoscapes might build on. Thanks.
Great. Yeah, thank you, Liz, for bringing that up. I completely agree that this is very much a trace element centric view. And so apologies for that, and I'll blame Phil as well. But I think the challenge for us as a community is to then identify the places where we can't get to our objectives without organics. I mean, we need an organic geochemistry and characterization, and I know there are a number of those. And I think just as GeoTraces has taught us that the entire periodic table is not relevant for the biological processes and the biogeochemistry that we're trying to constrain here. There are only certain elements that we think have controlling roles. I think we need to figure out what the geochemical space is that is required to answer our questions. And that's a challenge to the trace metal folks to understand that and to bring that in, and to challenge us as a community. And I think to the organic community to point that out to us. Say, listen, like there's no way you're going to get at these symbioses without infochemical abundances or something, or metabolomics. That's how I view it. I really welcome, this is a moment for discussion, not just questions. I welcome other people's opinions, but I think I'm very aware that the BioGeoscapes, sort of the folks who have been pushing the ball so far, have primarily come from a GeoTraces background, and we need others to help push that ball, or maybe push it past where we've pushed it. And this is an opportunity for us to hopefully gather some of that support. So I welcome that critique sincerely, and I hope that you will help us figure out how to take it up and move it forward. Yeah, Bethany.
First of all, thanks for setting the stage in terms of that sort of, I really like that figure that you and Phil developed in terms of the landscape of where these projects fit, and we can all put other projects
right. Yeah,
on
I
there,
sort of
right?
meant to say there's a lot missing from there.
It was a helpful framework to think about, and something that's really changed in my thinking about this program is thinking about these sort of survey approaches that Geoscapes has really been at the forefront of. And what's really, I think, emerging as we've had these conversations around multiple working groups, and you really highlighted it with the Bermuda data, is the time series component, repeat occupation, time series, the types of, especially from the biological side, a lot of the data that's already collected and they're very international, right? We have time series all over the world. And so, maybe this is also thinking about bringing in more geochemistry to some of our time series that are already going on rather than spinning up a bunch of new surveys. And so that's something that I've been sort of thinking about over the last day or so is, wow, we've got, other kinds of baseline data that, the Bermuda example I thought really captured that well. So just thinking about that.
Yeah. Thank you. And thinking about whether we need the same thing for, do time series have different components of geochemistry that added or different? Well, yeah, the same.
Okay, thanks a lot. Thanks again, Ben,
Thank you
for this nice presentation. So we will move south for the next presentation to the Centro Universitario Regional del Este. Cecilia Alonso, talking about time series. Picking up on the last question.
Good morning, everybody. I would like to start thanking the organizers for setting up this wonderful meeting and giving me the chance of being here. And second, I need to apologize for my rough English. So today I'm going to talk about a project that we are running in collaboration with the MPI for Marine Microbiology. And this is the implementation session. I was requested to focus on the challenges that we are facing to carry on this research. So I will first introduce you about the rationale of our project, show you a bit of the results, and at the end talk more about the challenges. Okay, so the main goal of our project is to compare taxonomy-based versus trait-based models, trying to predict different processes of the carbon cycle. We are located in here. This is, sorry, I'm getting messed with that. So this is Uruguay, between Brazil and Argentina. We are located in here. Our site is situated in a marine protected area. The observatory was launched in the frame of the Ocean Sampling Day initiative. And more generally, this region is a highly productive region sustaining large fisheries. And we are having several fronts occurring there. We have the Rio de la Plata estuary, which is one of the world's largest estuaries. And of course, there are all these other fronts, the shelf subtropical front and the slope. And there is also where the Brazilian currents and the Malvinas currents are, it's the confluence of these currents, and in front of here, the subtropical convergence is happening. And on top of that, the very site where we are located is experiencing seasonal upwelling in summers due to the winds. And this site is also located at the edge of 3 ecoregions of the marine ecoregions for the coastal zones. And it has been detected as a hotspot for ocean warming. So we really think that it's very nice to look at the communities and processes there in this so-called space for time substitution approach to trying to forecast what could happen in the global ocean. So what we are doing there is that since 2018, we are sampling regularly there. We already have like almost 80 sampling campaigns. When we go there, we measure some in situ variables with the CTD, but also the pH, the oxygen, the fluorescence, the turbidity, and some more. The wind velocity, we are also measuring the CO2 flux. And then back in the lab, we done the macronutrients, DOC, and some characterization of the DOM quality by spectral properties, HPLC, and now probably we can add some mass spectrometry there. We are also measuring processes, primary production, respiration, bacterial biomass production, and we are running experiments for the bacterial degradation of DOM. And of course, this, in fact, our main focus, my main background comes from the communities, so we are using flow cytometry for analyzing the picoplankton abundance and the taxonomic characterization of the prokaryotes and the protists using amplicons for the ribosomal genes, and metagenomics for the prokaryotes, And we are also running these old-fashioned things with microscopy for identifying the phytoplankton, the microzooplankton, and I'm still keeping filters for the prokaryotes because I'm a fish person, catfish person. So I hope that we will make good use for them. So I will show you just a few results because the whole project was relying on the theoretical expectation that we would have recurrent dynamics there because of the seasonality, and fortunately we found them. So we found 2 recurrent phytoplankton blooms occurring there, one in late winter by dinoflagellates and another in late spring and summer by diatoms. And we also found a recurrent pattern in the pico-autotrophic plankton, especially for the picocyanobacteria. They are the ones that are in cyan, that they are picking mainly in spring. And we also found recurrent patterns for the bacterioplankton composition. Here in this graph, in the y-axis, is the distance, the bright Cortis distance for the 16S data a long time. And we can see that there is this, I'm sorry, I cannot find it here. Better this one. We can see this is 1 year. These are 2 years, and what we see is that we find this shape where the communities are more similar when they have 1 year difference. This is for the 16S, and this is for the cassines. We find the same trend. And these are the processes, and this is again something very old-fashioned. We are running incubations for the DOM degradation with the water, which is pre-filtered and then incubated at in situ temperature and dark. And we measure the consumption of oxygen continuously using optops. And then we are also doing bacterial biomass production with the tritiated leucine incorporation, and we're measuring the decay, and the primary production and respiration using the clear dark bottle approach, also incubating at in situ temperature and the photoperiod of the day, and evaluating the oxygen with Winkler. Very old things. And now I'm going to show you the first preliminary results of our first attempt. This is preliminary and incomplete, but I just wanted to show you our approaches of what we are doing for modeling these processes. So for the DOM, based on the oxygen consumption, we could distinguish among 4 different categories of experiments, from the low to the high consumption. And what we did is we ran an ordination analysis for the 16S data and for the cassines data. And then we took the axis of this ordination analysis as variables to formulate models, multinomial regression models were in this case, in order to see whether we could predict into which category each of these experiments was falling. And as these are the confusion matrices derived from the Lib1 outcross validation, and as you can see, we have very high prediction. But of course, as I told you, these are very few experiments and preliminary, but gives us hope that maybe when we finish that set, we will find something there. And I also wanted to show you the primary production and respiration. Here I put the pattern, the results, the raw results, because I think it's something that was interesting is that, of course, this is the gross primary production, respiration, and net primary production. Of course, we see these peaks linked to the phytoplankton blooms, but we also found these situations here where the net primary production was negative. This means that the respiration was higher, and this was linked to a bloom of heterotrophic dinoflagellate, which is normally not occurring there. It's distributed up north, but there was a temperature anomaly at this time, and we found that, and I think it's interesting. To finish with that, for this we took another approach, formulating generalized linear models, and the values that we obtained were very close to, the predicted values were very close to the observed values. And I'm coming to the end with this. With the challenges, there are some things that happen over the world, and there are some other things that I think they are more particular for the southern part of the world, for the global south, and these are the challenges that maybe we can try to solve by participating in an international initiative. Of course, we have the pandemic, and for instance, we had to turn into the diagnosis lab. For 2 years, we were doing SARS-CoV analysis. It is very difficult to maintain stable teams because of the location, because of the lack of funds. It's very difficult to cover all the areas that are involved in this project. We lack trained people. We can, for instance, take samples, measure them in some of the equipment that we have, others we don't have, but sometimes the analysis and the interpretation of these results are difficult because we don't have, for instance, chemists that are interested in the organic matter of the ocean. The sampling logistics is complicated because we don't have our own ship. We depend on a third party, and it's difficult along so much time to keep the focus and to adjust the logistics according to the to the different changes that we're experiencing. And also something else that is the last thing there is the batch effect, what we call the batch effect for the analysis, is that as we don't have access to some of the equipment that we need to evaluate the samples when we have collected after a long while, and sometimes then when you get the results, you see that some things went wrong and you cannot fix them anymore, sometimes yes, sometimes not. But I still think that I'm committed to that, and I will keep on doing this. And I think that it's worth a while to have a global network of observatories, including the global south of the planet. And this observatory belongs to a broader network of observatories that we recently established in the frame of this Micro-SUDAQUA network. That is a collaborative network for aquatic microbial ecology in Latin America. So sorry that I was going over the time. I just want to thank the people and the agencies that make this possible. Thank you for your attention. Sorry.
I guess this is more of a comment than a question, that the amount of work you've accomplished is incredibly impressive in terms of a lot of those measurements that are not often routinely measured in these time series because they're so labor intensive. And again, that sort of gets to the point of really leveraging those time consuming, for the eukaryotes, if you're actually counting microscopy, that's an absolute number. And we have a really hard time getting there from omics. So incredibly valuable data. So thanks.
Thank you. Thank you very much.
Yeah, Lois Menier from Brest University. First, I want to go in the same direction. I'm running the Brest Bay Observatory, and I really want to acknowledge that it's a huge amount of work to run such observatory, so congrats for this. And I was wondering, so you did all these metagenomics measurements, and did you see, or did you start to observe changes in gene content, community structures, and mutations accumulating during this? I mean, could you start doing some real-time evolution of your microbes with this data, and like mutations that were fixed or genes that were increased in abundance, or is it something that you didn't start observing?
Yeah, we haven't looked at that because the main focus, now we are trying to close with this project, and the main focus was to use this data like as predictive variables only. But there is someone at our group who is very interested in the evolutionary aspects of it and more into the real ecology of it. So there is a chance for looking at that. There is a chance. We still have to finish with the sequencing, but yes.
I have maybe a question if nobody else. Thanks a lot, Cecilia, for this presentation. So you talked about the network. Mm-hmm. Can you just recall us how many stations or labs are participating? And did you do already some exercise of intercalibrating the methods that you're using in this network?
Yes. So the network was recently established, and most of the sites are freshwater, are continental, but we do have, I think it's 4 or 5 sites which are marine, starting from Costa Rica to the Beagle Channel. And we did have several discussions on the methods, and we are just about to produce a protocol for methods that would be for what we need to measure at every observatory. Of course, we will benefit much from the interaction with you all into improving this method and to make it more comparable to what other people is doing. Yes.
So thank you so much. And this got me really excited because for the last 15 years, I've been trying to develop a burgeoning network of global ocean time series and focusing mostly on ship-based time series that are making these kinds of measurements, including these labor-intensive rate measurements and the microscopy. And we held a workshop in 2012 in Bermuda focused on methods intercomparison. We are holding another workshop in January of this year or next year, and I completely invite you to come at least talk to me about it. But we would love to talk more with you about trying to, this one will focus on fair data practices, but we also are going to revisit some of the methods work that we did a decade ago in 2012 because methods are eternally evolving. But I would love to talk to you more, and we really want to have more engagement from the Global South, and the types of time series you are running are exactly what we're trying to bring those practitioners together to identify common methodologies and data structures that can help all of us. So thank you.
Gladly. Thanks to you.
Okay, we will then move on. Thanks again, Cecilia. So we're staying on the rate measurements. There's not more announced on the title for the moment. And it's Erin Bertrand from Dalhousie University who will give the next presentation.
Can you hear me? Can you hear me okay? Okay, great. Great. So I think we've heard a fair bit in the last few days about this idea that we want to be able to translate between omics and all the information about physiological potential and function that we have from that omics work into quantifiable rates and ultimately biogeochemical processes and their representation in Earth system models. And so I'm here to tell you about some good news in that we've gotten we've gotten a new SCORE working group funded to do just this. And the aim of this new SCORE working group is to develop a community and a framework for co-designing with people who do physiology, people who do omics, and people who do modeling currency converters to link omics data sets and biogeochemical models. And we're calling this PRIMO, Physiology and Rates in Microbial Oceanography. So this is an officially funded SCORE working group. And Robert Strepik and myself are the co-chairs for that. And there was some heavy lifting to get this together and funded by Maite Maldonado and Kaya Zervigil and more. So I'm excited to be able to share with you some of the terms of reference and the aims for this. And we developed this really in recognition of this major gap and this major thrust that BioGeoScape has to connect all of these different skills through common currencies. So the goal of this working group is to set the stage for development of a core suite of measurements that are harmonized, that are low cost, that are easy to use, and can be high throughput, that will link omics, rates, and biogeochemistry. We're thinking about things like new physiological measurements. We're thinking about new ways to combine omics measurements and either cellular modeling or some statistical modeling. We've heard some really great examples from Cecilia and from Scott yesterday about how these statistical methods can also help us to estimate physiological rates, and also new ways to directly estimate rates from omics measurements. And we have the vision that this is, of course, best if implemented at scale and at low cost for very broad accessibility. So these are the ideas here. In terms of reference, first we would like to review the current physiological metrics, identify gaps, and rank candidate assays. We want to identify physiological metrics for marine microbial metabolism that's best suited to convert between currencies of omics and models. We've heard some suggestions already in this meeting so far. We plan to make a bigger list and evaluate that. We also would like to determine the path, a useful pathway to assay development and build a framework and a toolkit that lots more folks can use to develop discovering proxies of physiological processes. And we also hope to identify a flagship physiological metric to develop assays that can be made high throughput. Those are our terms of reference and some key activities I'd like to draw your attention to. First, we hope to develop an open access database of current and desired physiological metrics, and something I'd like to call the Ocean Metabolism by the Numbers database with key physiological parameters that would help us all in delivering these things. We would also like to deliver a web-based assay workflow optimization tool, so something that people can use to implement their, the assay that they're most interested in developing. We also plan a mentorship program and a hybrid training course. And most importantly, we need the involvement of many more people than are actually on the list of the participants in this SCORE working group, and we're hoping that you will all want to be involved in this. So the ways that we're planning for more broad involvement are bi-monthly workshops that are open to all, and we'll make sure that that gets disseminated to the Biogeoscapes community and beyond. And we're also planning town halls and open meetings at upcoming conferences. So this is recent news for us. We're very excited, and there'll be more detailed plans forthcoming, but be on the lookout for, in particular, these bi-monthly workshops that are open to all. I wanted to take this opportunity to give you one quick example from work in my own group where we're trying to determine whether we can develop a protein-based approach for estimating primary productivity. So we're focused on this protein here, rubisco. Of course, we know that it's often the rate-limiting step in carbon fixation. It's often CO2 saturated in phytoplankton. So our hypothesis is that under certain conditions, rubisco concentrations can be used to estimate carbon fixation rates. So the way that we go about measuring rubisco concentrations is in a taxon-specific manner. We, at sea, filter water, preserve it, or freeze it. In a lab, we extract that protein and then we apply a mass spectrometry-based approach, in this case, targeted mass spectrometry. And we've done some analysis to identify specific peptides from rubisco that are diagnostic of groups of particular importance in the environments that my group tends to focus on, the high-latitude environments. So Phaeocystis, diatoms, green algae, and we've identified peptides that are broadly applicable or broadly distributed amongst most eukaryotic rubisco sequences. So then we developed these quantitative methods to be able to determine moles per liter of rubisco. We've developed these quantitative methods to measure moles per liter of rubisco in seawater. And I'm going to show you one quick example of how we've applied this. So this was a cruise in the Labrador Sea in May 2020, and was meant to be examining the biological carbon pump when we encountered the decline of a Phaeocystis bloom. And I'm showing you depth here, if you can see the mouse, depth and peptide concentration of specific rubisco peptides. So peptides from haftophytes, so from Phaeocystis in this case, and then several different groups of diatoms and Micromonas. And what you can see here is that we have a dominance of Phaeocystis, like we expected based on the smell on the boat. And this is work done by my PhD student, Scott Polera. So we're excited about this. This tells us that there is a lot of Phaeocystis present. It is contributing to primary production potential. What we wanted to do now was evaluate whether we can convert these concentrations into rates in a useful way. So we made some assumptions. sorry. We make some assumptions to do this. So we assume if CO2 is saturated, we can use the specific activity of Rubisco to calculate carbon fixation, and specific activity of Rubisco and our Rubisco concentrations to calculate carbon fixation according to this simple calculation here. But what this means is that we really need to know this KCAT value, the specific activity. So what do we know about Rubisco specific activity? Here's a plot of KCAT Rubisco specific activity versus temperature. And the lines are fits to data with using an Arrhenius curve, and the dots are actual measurements of data points. And so what you can see here is that we have a very large range of KCATs at higher temperatures. But as we get to lower and lower temperatures, these rates converge into a somewhat manageable range here. So we hypothesize that we can apply the small range of KCAT values at low temperature to reasonably estimate carbon fixation rates. I'll point out that there's very limited biochemistry available here, so we're making some assumptions, but we think that those assumptions might be good at low temperature. So if we combine these things and make our Rubisco measurements, and we use this conversion, we compare to C13 bicarbonate uptake incubations, and I'm showing you here the uptake rate incubations in blue and the Rubisco-based estimates in red over 3 depth profiles in the Labrador Sea, and we see what I was surprised to find, that there's remarkable coherence in these estimates and the rate measurements from bottle incubations here. So you can see there's also some really interesting differences that we're interested in exploring, but in general, this is a much better estimate, or much better coherence in these rate measurements than I was expecting, and is promising, I think, for us to identify additional candidate metabolisms that can be assessed using these kinds of rate measurements, and combine that with the promising work from the statistical modeling that we saw yesterday and today. I think we might be in better shape than we thought, especially when we combine this with all the brain power in this room and the SCORE working group that we were recently funded to do. So in summary, I think Rubisco concentrations show promise as a metric for quantifying primary producers, for examining controls on primary productivity, and potentially for estimating primary productivity under certain conditions. Our next steps, I think we need more biochemistry. We've been saying this all week. The same is true for this particular assay. And I think it'll be really interesting to explore comparisons of this data with additional productivity metrics in the field. Just some acknowledgements here, some contact information for those of us who are leading the SCORE working group, and thanks all for your attention.
Thank you very much, Erin.
Adrian Marchetti from UNC. Very nice talk, Erin.
Thanks, Adrian.
So your protein-based primary productivity, are you assuming it's more gross or net? Probably more gross, right?
Well, I was assuming gross, and then
so the C13 24-hour is a net primary productivity.
Yeah. So we've got a lot more investigations to do, but I think this is more data, but we were assuming gross. It looks like it's closer to net in this case, but I think there's more to be explored here, and I think that's really exciting.
So have you thought of a way maybe to estimate autotrophic respiration through proteins?
We're working on it. I kind of like Scott's proposed method of statistical approach there, because I think that's really complicated. It's harder to identify rate limiting or potentially rate limiting enzymes in that case, but there's lots more to talk about there, and that's why I'm excited about this. Yeah. Thanks, Adrian.
Julie Granger, UConn. Great talk. I was wondering, I anticipated that at some point they would no longer be correlated because you get light limited, right? And
me too, yeah.
Okay.
So I'm excited to explore the hypothesis that there's a feedback between light availability and Rubisco production that helps explain that, but hypothesis now. Hi, Kai.
Now it's working. Fantastic. All right, Kai Tiafogo from the University of New Hampshire. Thank you very much. So, quick question: how do you explain the temperature dependence? So that's not that your method is not working at higher temperatures.
Oh, I mean, so there's temperature constrains these rates to a large degree, and there's still a range of rates or of specific activities at low temperature, but that range just, it converges. Yeah, we also obviously need more data at that low end too, so we're making some assumptions here. But yeah, this general property of enzymatic.
Thanks.
Yeah.
Thanks. That was really cool. How is Rubisco like a Goldilocks and very unique in that it has this not substrate saturated situation? I've noticed. Are we likely to find that there's many other proteins in that situation?
I think it would be a really fun exercise to categorize enzymes into potentially substrate saturated. And yeah, this is a community exercise that I would love for us to do potentially as part of that SCORE working group. I think there are more. I'm not going to make a list now, but I think there's potential. Especially, it'll help us to sort. That's one of the objectives of the first. One of the first aims of the working group.
Hi, Shane from Shamir University. As you compare with the carbon 13, so on 24 hours results, as you see the results is comparable, that means the PDOC path is not important, since that is PPOC, right? The production of the dissolved organic carbon.
Yeah, I mean, there are clear gaps in these. There's clear differences here. So I'm really interested to explore when that is important. And I think there's ways to couple some of these process studies and these omics-based assessments to ask questions like that, which I would be really excited to do. Yeah.
Okay, thank you.
Last.
Hi, I'm Eric Webb from University of Southern California. First of all, wow, right? But May 24 to May 30, speculate on the differences.
We were nominally observing the decline of the phases just bloom there. I don't know. We're working with our physical oceanographer colleagues to try to understand water mass movement here. There's some complicated physics happening in our nominal time series. I don't know yet.
And do you think you're missing certain Rubiscos, and that's why you're getting?
Maybe. It was so phasicist dominated, but I don't want to write that off. And we're doing, so this is very targeted measurements now. We're coupling this we're processing metatranscriptomic samples now to try to see if there's a diversity of Rubiscos that we didn't identify with these quantitative measures. But yeah, I'm going to say physics.
Thank you very much. Excellent.
I'll make use of my clock is ticking, so I'm going to start talking. So I'm here to talk a little bit about what was labeled originally as a kind of modeling talk, but I think what I'll try to do in these kind of 10 minutes, talk a little bit about how we can kind of try and scale up our observations and the tools we've got to look at global change and to give some sort of context and framing for the role models might play in doing this. I know. Okay, so I want to talk about this grand challenge we have that's beyond this room, okay, and connects to how biogeoscapes might be interesting to society, the guy at the Mexican restaurant and things like that, which is understanding how the productivity of the seas is going to change, how it's going to respond to the global change, all the perturbations we're exposing the ocean to. And of course, we think that marine microbes are playing a really important we all think that marine microbes, I'm not going to talk about rhodopsin now because of that, Adrian. There was a load of slides on that, but they're gone now. And we think that marine microbes are playing a really critical role in responding to this challenge, but they're doing so at a range of different scales, right? down at the intracellular scale, up at the organismal assemblage scale, and then ultimately integrating things up to the scale of the ecosystem, where we get all these interesting ecosystem scale cycling and feedbacks. We want to understand how the physical environment is going to change, right? And the tools that we're going to use for this, or that we do use for this, are Earth system models exposed to different types of climate change projections. Now, this is the sort of state of the art for SST, sea surface temperature, and on the left-hand panel there, you can see the global average sea surface temperature normalized to some pre-industrial baseline state. The different lines focus on the solid lines. They're different CMIP6 projections, shared socioeconomic pathways. The red ones are the high emission scenarios. The blue and green are these, unfortunately, ambitious mitigation pathways that at some point we hope we're going to end up on. Okay, and what you can see is when you look at the projections that come out of all of these Earth system models, we can really clearly see the difference between the red line and the green line and the blue line. All these modeling groups, they give really well-constrained projections of what the global scale change in sea surface temperature looks like. They can produce maps at regional scale. Here I've pulled out the high emission scenario and the SP126, which is the blue line on the left-hand panel. And these dots represent what is the sort of classic way of trying to discriminate amongst Earth system models, which is, you'll think this is quite a low bar, but it's whether they agree on the sign of the change, right? And okay, so if there's a mark here, they all agree, unsurprisingly, the ocean is going to get warmer. And we understand this very, very well. Okay, the utility of this skill evaluation metric will become important or relevant on the next slide. Okay, this is net primary production. Okay, this is something that we think is this critical ecosystem flux, all right? And on the left-hand side, you see the same thing as you saw for SST. You're looking at the, the global change in percentage terms compared to a reference period back here. And now suddenly you've got this whole mash of variability, right? If you look at the previous slide, we can really clearly see SST. This is really clear information to communicate to policymakers about how different choices we make, about our lifestyles, our carbon demand impact on environmental change. We're not in a position to give that kind of information for net primary production, right? You might believe it's decreasing in the highest emission scenario. But when you're giving a kind of multi-model mean projection of 0 plus or minus an enormous number, that starts to become incredibly less useful. It makes it look like we don't know what's going on, okay? At regional scale, these differences become even bigger, right? So this same as for SST, high emission scenario, low emission scenario. Now just focus on how little of the ocean gets a dot, okay? That dot, that absence of that dot means that the models don't even agree on the sign of the change, okay? That's a massive disagreement, okay? And that's a big issue. We can do these same types of analysis for things like for other rates, nitrogen fixation. We've done these kind of studies. Similar, enormous uncertainty across models. And you might even actually believe that all these models that we plot here, they're actually quite similar in terms of how they're set up and what they try to do. So I don't even believe this ensemble represents even the real uncertainty, right? If we actually had a broader, more representative group of models, you might imagine that error bar would be even bigger, okay? So this is a big challenge. I'm not going to, dearie me. Okay, at the regional scale, these numbers are even bigger, right? Well, we'll get there eventually. There we go. I don't want to spend too much time on this, but this is, we were here, right? This is the global scale. Just look at the width of that uncertainty bar. That's 1 sigma. Okay, and this is the same quantity plotted for all these different ocean provinces. Notice how much bigger the bar becomes, okay? So at regional scale, we've got an even bigger problem, right? And it's at regional scales where people want to use this information, right, to project what's going to happen to a fishery or this, that, and the other, okay? So we need to get engaged with this. We need to get engaged with this because people rely on the sea for where they get their protein, largely. Okay, these are projections of the change in the maximum catch potential in exclusive economic zones. All these little colors, that's basically, in the absence of fishing, how much biomass might you be able to extract from the ocean through fishing. All the blue areas are where that catch potential is projected to go down. Okay, you see most of the ocean bar, a few high latitude regions, is blue. The colors inside the national frontiers are basically an estimate of how much, what proportion of the protein intake in those nations comes from the sea. Okay, so you see all those places. Let's just zoom in here or here, all these places, all these blue numbers are places where society relies on getting their protein from the sea. So this is a big deal, right? I think we need to think about how what we're doing in this room that we're all super excited about might actually reach outside this room and respond to this big challenge. And in part, maybe it links really nicely to what you're talking about, Erin, about how can we maybe harness this fantastic data that we're all talking about to improve the models and reduce uncertainty, okay? And now I would normally tell you a lot about these different types of models that we have that you can read on the screen. This is our toolbox for responding to kind of global scale environmental change. I don't have time to do that. There'll be a modeling meeting next year where we can really get stuck into some of these issues. What I want to talk to you about is a little bit about how these different types of models are trying to push the envelope a little bit to take advantage of the fact that we know that they're really simplifying the environmental stressor landscape. They're simplifying the degree of biodiversity they represent. They're simplifying the degree of phenotypic plasticity, resilience, adaptation, acclimation that's going on, right? So how are they trying to deal with these things? Let's start with these global biogeochemical, it does work, global biogeochemical models that are embedded within these Earth system models. So these take this kind of like PFT-based approach. I've shamelessly just used one of my own examples, but Nick Halko, University of Hawaii, is the lead author. So this is a representation of this kind of model, right? We have these coarse-grained pools, and we try to deterministically model the transfer of material between these pools, and some of those material transfers represent then key fluxes like net primary production or export production that we're interested in, or nitrogen fixation as well. Generally, they have a very small group of nutrients here, normally nitrogen, phosphorus, silica, and iron. But we know that there's lots of emerging evidence that other micronutrients, things like that might be important. So one way we try to address this challenge is we just complexify the tool, right? We add extra boxes, and you can see your favorite element, cobalt's there, MAC, in the mix. And we add these elements, right? And then we try to model their flux through this ecosystem. And that allows us potentially to say, okay, well, how important is manganese limitation in the Southern Ocean, potentially from a modeling standpoint? Bigger the bar, more limiting. And that's nice. We can use that, and then we can put that through the same system modeling workflow that we have to see how important might that be, how much extra uncertainty might that add to the projections. But this is never going to deliver at the end on these challenges, because this is still an incredibly simplistic representation of the ecosystem, okay? It can test specific hypotheses, but we're never going to be able to lump in all this richness of data that we're talking about in this room. That's led to the generation of these kind of statistical models, right? You might call them machine learning. I tend to just call them statistical multivariate models. Paul showed some examples of that on the first day, using the TARA data at a great degree of granularity. I've chosen a different example here because it's a super simple one. You try and build a model for the abundance of Prochlorococcus and relate that to sea surface temperature, right? Flombaum showed this, and then you take the projection of sea surface temperature, which, as we saw at the start of the presentation, is done with very high skill, and then you say, okay, I'm going to use my statistical relationship to show you how climate change is going to affect the abundance of Prochlorococcus in the ocean, okay? That's very powerful. You can address biodiversity questions. You can do this for other types of key groups that you're interested in, and that presents really detailed and really rich information. Of course, the problem is that it assumes that the relationship between the predictor and the determinant kind of stays the same in the future, which I'd argue remains to be seen, let's say. And this is a big issue in terms of also the fact that you assume that the sort of range of environmental conditions that we see today in the ocean are also there in the future. How do you deal with novel conditions? We know the future ocean is going to be much lower pH. Do we find those conditions today? So that's a drawback, a challenge for those kind of statistical models. Ultimately, we might think of these metabolic or genome scale models, which are these really leveraging, by definition, the rich genomic data that comes out of programs like TARA and others in this room, where we try to look at the flow of metabolites and metabolic reactions within the cell. We can force this metabolic model with things like light, nitrogen uptake, phosphorus uptake that comes from an Earth system model, right? And we can just completely remove all the growth rate calculations and allow this metabolic model to produce a prediction of growth rate. That's what it looks like globally, okay? It works. We can do this as an engineering sense. And that's really exciting because now we've taken all the guesswork out of the parameterization of the peak of phytoplankton growth rate. We can also then learn other things that we definitely don't include in our Earth system models. Here's an estimate of the glycogen production associated with this metabolic model. And so we can really mine the kind of metabolic richness here. The challenge here, of course, is that this is a prokaryote. We have the metabolic model for pro. We don't have them for eukaryotes. So ultimately, they're not, we need to do work there on developing these metabolic models so we can use them in our pipeline. Okay, we want to do this, right? We love these kind of things. We want to go around a wheel and generate new observations, new understanding. I'd really say here, this understanding is what we need in the modeling world. You can think back at that. To finish, I'll just put this up. I think, if we want to deliver on these big societal questions, we've got to use models. They're the only ways we're going to be able to make projections that link social choices to impacts in the environment. And we have the potential in this room to contribute to the reduction of uncertainties, and that's a really important driver for this program. There's different types of modeling, and what we need to do actually is identify ways we can make progress in the next 5 to 10 years by leveraging these different tools, leveraging the different types of data to demonstrate actually the utility of this program. I'll stop there. Thanks for listening.
Hello. Al, that was a very nice way of simplifying what various models.
I didn't say anything.
That was nice because it made me think about why should a model be based only on one thing? Can you not make hybrid models?
Good question, a good point. In the short term, certainly, the solution is to leverage, to use the 3 different types of models maybe for what they're good for. So for instance, you might use a metabolic model, which is very costly to run, to maybe find statistical solutions or parameterizations for key processes that you might ultimately then embed in your Earth system model, which is what we kind of do now, except at least in that way, we'd have a strong sort of mechanistic basis for those kinds of parameterizations. So absolutely, we need to think about, I think, instead of it's not a binary choice between one or the other, actually. I think we need to work together much more to use the different tools where they're strong, I think. Ultimately, you're always going to have to use an Earth system model because that's how you link emissions, radiative forcing to the ocean. So the key question is the role for statistics, machine learning, and the metabolic modeling, how that can handshake with those tools. Thanks.
Hi, Al. Liz Kuczynski from WHOI. I have a question about sort of the approach of biogeoscapes towards some of these larger questions. And very specifically, I was wondering if you could provide some thoughts or speculations on where biogeoscapes might want to think about placing itself relative to some of the conversations around carbon dioxide removal in the marine environment and this emerging industrial commercialization approach. I guess just to provide my personal feeling on it, I'm somewhat concerned that if we're not part of the conversation, the need is so urgent across the planet that it will proceed without our input if we're not ready to provide it. So, but I'm torn on that approach, and I'm not entirely convinced that that's the right one. So I'm just curious if you've had some thoughts on that, and if you'd care to speculate on how to connect in with some of that work, especially from the model.
I've never thought about it. And I only have about 3 conversations a day, apparently, at this meeting with different, and we haven't had one yet, so we can start having it now, right, Liz? It's a tough question to answer, right? Because, I've got my personal view, and then there's what we might want to actually do as a program. There's no doubt it's got to be a key driver for the rationale for what we're trying to do, because we can provide through that the kind of guardrails for how other people are doing this kind of activity. Now, how much we want to get involved in shaping that then becomes a personal choice, ethical choice, moral choice even. I think at the least we've got to be lending our expert judgment and our expert assessment of what's going on, because we do have a lot of expertise in this room, and many of the CDR efforts, either by design or implication, end up affecting microbial communities. So I think we can't ignore it. You're absolutely right. We'll have a beer later and talk more about it.
Al, thank you very much. Jamie Becker, Alvernia. I think it's a naive question, but in the beginning, the discrepancy between the black dots in your temperature maps and your NPP maps, is that primarily due to undersampling, that discrepancy? Is it primarily due to comparing an abiotic parameter to a biotic parameter that's a summation of a lot of abiotic parameters?
You mean the NPP projections and the lack of agreement?
You mentioned like the lack of black dots there, meaning the models don't even agree on the sign.
Yeah, that means
why there's so much less. Is it undersampling? Is it biotic versus abiotic complexity?
Well, what it says is that we don't have, if we had a clear mechanistic understanding of what was regulating primary production, there would be very little room for the modelers to be creative in how this is parameterized, and it would look a lot more like SST, right, where there is a rock-solid theory, there are laws, there's the Navier-Stokes equations. There's not much creativity. Turbulence closure maybe is a key challenge for system models, but it's largely about resolution, the physics, right? High resolution, more accuracy. For net primary production, as we discussed, we even sort of somehow want to understand how, what the different measurements even mean in terms of net primary production. We don't have a clear constraint on the models, okay? And that means that there's a great liberty, it's a very inclusive kind of community, that you can set up your Earth system model in the pre-industrial state to have, globally I'm talking here, 20 petagrams of carbon being fixed every year. Other Earth system models have over 100 being fixed. So there's a factor 5 uncertainty, and even the starting point. That's why we normalize all the projections to the mean, what we call the mean state, because otherwise it would just be completely wacky. So I think this is a big issue. We don't understand the magnitude and the processes well enough as a community to place enough constraints on the models, and that's why they disagree, even on this simple metric of skill.
Thank you.
We have 2 online questions.
Oh, great.
I was going to ask Jim Moffitt, did you want to unmute and ask the question, or would you like me to ask it for you?
You can ask it for me. Thank you.
All right, don't shoot the messenger. Okay, since today's agenda is about implementation, how can existing models help us to choose where to sample?
Good morning, Jim. Hope you're well. Yes, so this is an age-old kind of question that we get, right, about how we can use models to guide sampling. And so I think, the classic answer would be that we can look at where things are uncertain, and we can go there to try and resolve uncertainty. And that, I think, is still a really good approach. But I'd still caution that a little bit, because the uncertainty is still always only going to be relative to whether you think you have an accurate model that represents all the processes of interest. And the example I'd use there is the Southern Ocean, where the CMIP-6 models actually agree incredibly well with one another. And it looks very certain, but we know, or we suspect, that these models are actually all completely wrong. They can't reproduce current trends, et cetera, et cetera. So I think estimates of uncertainty, I think if you want to look at different regions, Jim, you've got to then look at leveraging regional models, which can then be more complex. You can look at individual-based models, and it really depends on the question. I don't think there's a simple answer here, unfortunately, Jim. But it requires that dialogue between the observational scientist and the modeler to identify the questions together such that then we know the appropriate horse to run over that course. I'm sorry, Jim, I haven't given a good answer there.
Okay, I have an additional, more of a comment than a question, it looks like, from Fanny Montero. Fanny, did you want to unmute and make the comment yourself?
Yeah, sure. Hi, Al.
Hey,
thank
Fanny.
you for a good presentation. I think you gave a very nice overview of the modeling state and the issues. One thing I wanted to comment on is you were arguing that we need better understanding to improve the model uncertainties. And I would agree with that. The other thing I would argue also is that we need more observations, because we still don't have enough good coverage, spatially and temporally, to validate our current models. And we do need more observations, especially in places where we don't have, much coverage, so that we could improve our validation for process space, also build better statistical models. Thank
you, Fanny. Good afternoon. I completely agree. It links actually a little bit to Jamie's question. Part of that uncertainty, part of improving that observational constraint is through observations, right? That lead to observations in experiment that lead to improved understanding. So I completely concur with your view.
Thanks a lot, Al. Yeah, c'est fini. Thanks to all the speakers for the exciting presentations. And for keeping track of time. We did a very good job. So we go for coffee now. We meet 11:45 here. And I got information from Mac. At 12:30, we meet outside for the picture, right? Just outside on the veranda. Okay, see you 11:45 back here.
Shall we start? We shall start. Come on. Yeah. All right. So what I'm going to do now is introduce the next stage of this meeting and how we are going to go about this implementation effort. So I want to start with this slide from Mac's presentation, just to remind you that BioGeoScape is an interdisciplinary program, or we would like to be an interdisciplinary program by definition, where we hope to combine omics, biogeochemistry, physiology, and through modeling, understand and put it into the global context. As you've seen already, I think, we believe that this is going to be this is going to happen through multiple studies, ocean sections, transects, process studies, time series, and so on. And what is really important is that everything is rooted in intercalibration and data standards, so we can actually put all the data together and make sense of it. So this is from the AccelNet proposal. So we really care about omics and biogeochemistry, and in order to implement the program, we have to take advantage of data science, intercalibration, best practices, modeling, and of course, RATE to link omics to biogeochemical cycles. This is sort of what we want to do. We have spent Monday and Tuesday drafting the research questions of the program, trying to do that. I think you have done an amazing job, and we will come back to this tomorrow morning. But right now, we are going to focus on this. As I think I already said, there are various efforts right now looking at standard and intercalibration and bioinformatics. So we are going to focus on field sampling and technological developments, laboratory experiments and incubations, and modeling and prediction. So this is the way we have set up the breakout rooms. In essence, we have 5 of them. So one is going to focus on time. So what are the time scales of sampling that we would like to have, how we can use time series, using short versus long stations, temporal niches. So these are the things that you want to answer. How are we going to sample a space? Where are we going to go? To do what? So these are things you should be thinking about. Depth. How deep are we going to do? What tools we can use to enable more sampling at deeper depths. There's going to be also a group looking at laboratory tools and experiments. And I guess the question that we want to answer is how we can explore microbial sensitivity to environmental change to inform field campaigns, and what sort of laboratory studies you want to do. And then we have the models. So in essence, you're going to go to these breakout groups, and we don't want to prescribe these 4 questions as we did in the last themes, breakout groups. What we want you to do is, as you answer these questions, in black, consider these things, okay? So what is the overarching goal of BioGeoScape? What is feasible with what we have? How can we maybe enhance sampling by international collaboration? What do we still need to do? And how do we organize ourselves? So just think about this. I really love this. Ben made this, of course. And the idea, I love it. So yeah, you know, I live in Maine, and I couldn't believe in McDonald's they serve lobster rolls for $2. So anyway, so I think the idea that is coming out is that you can really be every nation can be part of biogeoscapes, and you might use many different things to do that. So this is what we call the franchise model. So you might be really involved into ocean basin scale expeditions, or you might really be involved in coastal observation, or you might have amazing time series that you want to be part of biogeoscape, or you might have amazing labs doing physiology, and that's going to be your contribution, or you might have amazing modeling. So the key is that we don't want to be we don't want to tell you what you have to do. Okay, prescriptive. We want everything to be driven by research, the research goals that we have set for the program. Okay, so if you are addressing any of the research goals that we set for the program, you can be part of the program, whatever approach you want to take. And of course, it's super important that we intercalibrate and we use data standards so we can intercompare all these global data sets. So I think that's the key. Now, I get this question all the time. What is a biogeoscape study? And the truth is, we do not know. But at the end of this meeting, we hope we know. So I'm just going to give you an example. I could imagine that a biogeoscape study will do some core parameter measurements that the modelers really want. Oxygen, temperature, salinity, chlorophyll, particular organic carbon, DOC, macronutrients. Everybody's comfortable measuring this, collecting these things, right? Trace metals. One second, no, I have a comment. Okay, don't be scared of trace metals. So I have a system that is ridiculous to call it a system because it's not a system. But I paid $3,000. $3,000, you can sample for trace metal cleanly down to 400 meters. And I guarantee you, every nation in the world can sample for trace metal down to 400 meters with this little system. Me and Mac and many others built a clean room in 2 hours in any boat. So there is a short string of trace metal sampling that I think everybody can do. So don't be afraid of trace metals, essential trace metals. And there are many labs in this room that are very happy to measure the essential trace metals. We're talking about 5, 6, 7 trace metals, okay? I will say there is some communities that are not ready for, they are not ready the way that maybe trace metal measurements are ready to be made. The organic chemists trying to characterize organic matter in the water, but we could collect samples for them and send them, and it's very easy, they tell me, collecting DOM, and if there is large volume sampling, a little piece of a filter, we could send them, and they will be very happy. So I think there is things that everybody knows that we can do, and there are things that we want to measure even though we are not ready, but in 3 years we might be ready, so we can start imagining that we are going to be able to do it. So this scenario, this idea will be any biogeoscape study will do this basic, what I call basic biogeochemistry measurements. And then you might want to really look at a research question and you want to use omics. or you want to combine omics and physiology. So we could say, as long as you maybe combine two of these and you do your core chemistry measurement, this is a bioescape study, So this is just an idea. So in order to answer this question, what we are going to do this afternoon is we are going to go into breakout rooms and we are going to conceptualize a bioescape study. So what will you measure and why? And we are going to look at the answers of these breakout groups, and then I think we will be ready to answer that question. And the idea is that by the end of the meeting, we can answer that. Yeah? So this is for you to, it's in the folder. And this is another thing you can keep in mind when you are in your breakout room this afternoon. Consider a common set of manipulation to set up core parameters to include in the franchise model. Adopt learning from proto-bioescape studies to each of the themes of the meeting. Identify the sweet spot where we can be on. Okay? So, last thing. Think big again. Okay? Identify challenges that can be solved. Don't be afraid to challenge yourself and your nation, and just, think big. Okay, so you can go to your breakout room. All right.